ಅಲಾರಾಮ್ ಬರ್ತಾ ಇರುತ್ತೆ. Good morning, students. So let us start the session after 2 minutes. Let us wait for a few more participants. If they attend, we will start. If not, we will start with 2 minutes. Okay. Please stay online.
Good morning, ma'am.
Hello. Good morning, guys. So few of them are new here. Yeah. And few of you did not attend the last session. You missed 2 sessions, I guess. So please be continuous, means please be what I say, consistent with your attendance regarding the sessions. Okay, so don't neglect it. Don't neglect any sessions. Not only biology, don't neglect any sessions, because certain questions which we were discussing, they may come for the exam. Okay, so we know that at the when the exams are near and when we prepare, so that preparation matters a lot that than when we prepare a month or a 2 months ago, right? So that is why attend the sessions and then practice whatever assignments will be given, whatever assignments will be sent to your mail ID. And you know very well that the students who attend the sessions till the end of the session, who attend the session till the end, and when they message us their mail IDs, when they message us their mail IDs, then we are going to send them the notes or the previous year question papers, whatever we discussed in the session. Okay, this is regarding how we will send the previous question papers or the notes. So many of you got, I guess, around 9 o'clock today. Janvi got, and I think Tasmia got, Sindhu got.
Yes, ma'am.
Please check your mail IDs if you did not get. I almost all the mails are delivered. I got the message from the committee that the messages or the mails have been already sent. Okay, so any of you you want the previous year question paper, please maintain your attendance till the end of a session. Then you have to mail your mail IDs. Those mail IDs will be forwarded to the respective member, and those members will be sending you the notes. Okay, right. So let us begin. So yesterday we started, once we discussed 2019 question paper, then we discussed the multiple choice questions on biomolecules. Today we are going to discuss a different kind of question. So we know that in biology, we will get the question based on the diagrams, right? So there are certain questions related to the diagrams which we are going to discuss today, right? Okay, so let us begin. Just check, can you able to see my screen? Yeah.
Yes, ma'am.
Yeah. So here this is a kind of question where we will be having the diagram related question and you have to give the answer. Okay. So here the second main, observe the following diagram and give answer. Okay. You have to observe this diagram. So when you see the diagram, we will come to know that it is related to, can you guess? What is the diagram you are seeing? What it is related to?
Replication.
Very good. Yeah, good. Yeah. So it is related to the replication of a DNA. So we will be having certain question based on that. Yesterday we discussed regarding the concept of replication also. The first question of diagram related question goes like this. Identify the following diagram. See here. Option A, replicating fork. Option B, ori. Option C, template strand. Option D, coding strand. So I want your answers through messages. So Janvi voted as A. Very good. The first answer or first question of today. Then Tasmia answered as option A, Sindhu, option A. Then what about others? Navya A, Smita A. Fine. Okay. Bhairish A, Shuchita A. Is this your first session, Shuchita?
Yes, ma'am.
Okay, welcome. Fine. So identify the following diagram: replicating fork, B ori, C template strand, and D coding strand. Almost all of you gave an correct answer. So correct answer is the replicating fork. Replicating fork is a right answer. Just a minute, I will just mark it. Yes, so replicating fork is a right answer. Okay, now give the term for starting point of replication. So from starting point of replication, what it is called as, you have to tell. Option A is ori, option B is ARS, option C is ROP, option D is SSB. Yes. Who is going to answer first? Yes, it is again Janvi, I guess. Option A, Tasmia, option A, Smita, option A, yes, Sindhu, option A. Fine. Okay. So the right answer, give the term for starting point of replication. So this is a starting point of replication where DNA polymerase third will act. From which point the DNA polymerase will act? So here the answer is option A, that is ori. Okay. Now give the term for DNA. Here I want the answer through the mic. So how you have to answer is, you have to tell your name. For example, Janvi A, like that. Tasmia B, like that you should say. Let us see who is going to answer first. Give the term for DNA fragments produced by discontinuous synthesizers. A, Sokezaki, segment B, Mokezaki, segment C, Okezaki, segment D, Zokezaki segment. Yes, who is going to answer first?
Tasmia Okazaki segment, ma'am.
Okay. Tasmia said, I heard the voice of Tasmia first. Janvi interrupted, but I heard the clear voice of Tasmia. So first answer is given by Tasmia. So she said Okazaki segment. Very good. And it is a right answer. Then the next question goes like this. Continue to answer in a mic, and all of you please attend and enjoy the session like this. Right. Fine. The next question goes like this. Give the name of enzyme which bind DNA fragments produced by discontinuous synthesis. Option A, DNA ligase. Option B, DNA polymerase. Option C, DNase. Option D, endonuclease. Yes, come on.
Tasmia A, ma'am.
DNA ligase, ma'am.
One more chance. Who is going to answer?
Tasmia A, ma'am.
Option A. Option A, ma'am.
Okay. Option A, DNA ligase is a right answer. Okay. The next question goes like this. In which stage of a cell cycle this structure is formed? That is, this structure means that diagram is formed. Option A, G1, option B, G2, option C, yes, option D, M.
Tasmia D, ma'am.
Option
Tasmia D, M phase. Okay. What about others?
Option B, ma'am.
Option B. What is your good name?
Sindhu.
Sindhu said option B. Yes. What about others?
Janavi D, ma'am.
Janavi D. Okay. D. M phase. Fine. Yes, what about others? Any other answers?
Option A, ma'am.
Option A, Tasmia.
Option D, ma'am. Tasmia.
Option? Yes, Tasmia, what is your answer? Kirtana and Bairesh, we are going to answer through a mic, okay? Not in a chat. I'm not checking the messages. Please do answer with a mic by your name and enjoy the session. So in which stage of cell cycle this structure is formed? GA, G1, B, G2, C, S phase, and D, M phase. Yes, last chance for everyone. Think and answer
Tasmia
fast.
D, ma'am.
Tasmia D. Yes.
Yes, ma'am.
So let me lock Tasmia's answer. Let me lock Janavi's answer. So she said B.
Option B, ma'am.
Option B. Okay.
Ruhina
Then
A.
okay. Who said A?
I said B,
Ruhina
ma'am.
A. Okay. Yeah, fine. Okay. Don't mind if I didn't take your name. Fine. Whichever I remember, I will take. Fine. The right answer is given by no one. So the right answer is option C, that is yes phase, synthesis phase. Here what happens in synthesis phase? In synthesis phase, all the proteins, the DNA fragments, all has to synthesize, all has to be formed. Replication is nothing but the formation of a new DNA, right? So it occurs in synthesis phase, that is yes, no one answered it. That is why I asked the question twice or thrice. Fine. Let us move to the next question. So here, observe the following diagram and give answer. Observe the following diagram carefully and give answer. Identify the following diagram. AEM picture, that is electromagnetic electromicroscope micrograph picture, bead in string. Option B, nucleosome. Option C, linker DNA. Option D, histone. Yes, any guesses? Yes,
Janavi A.
Jahnavi locked option A. What about others?
Sindhu B, ma'am.
Sindhu B. What about others? Yes. Bairesh, Navya, Ruhina, Shuchita, Smita, Tasmia. Any other answers? Okay. Janvi, you locked an option A, right?
Yes, ma'am.
Okay. And Sindhu said option
B.
B. Okay. Are you sure, Sindhu?
No, ma'am.
Okay. Are you confident, Janvi?
Yes, ma'am.
Okay, so the right answer is given by Janvi. Option A, it is nothing but bead in a string. So we don't have any nucleosome, we don't have any linker DNA, and D, histone. This is a common sense question. So the right answer is given by Janvi and Sindhu. Well tried. Okay. The next question goes like this. Give the term for regions in given structure which are loosely packed and stains lightly. So here, bead in a string. Before that, bead in a string is answer. Okay. But when we study this, we will consider this as a chromatin structure, thread-like structure for the next question, which is not there in this option. Okay. So let us consider this as the chromatin and tell me, give the term for regions in a given structure. Kirtana, can you mute yourself? Kirtana, mute yourself until I finish the question, please. Right. So give the term for regions in given structure which are loosely packed and stains lightly. Okay. Option A, heterochromatin, option B, euchromatin, option C, pseudochromatin, option D, none of the above. Which are loosely packed and stains lightly? Which kind of chromatin stains lightly? Yes.
Tasmia A, ma'am.
Option A.
Tasmia locked, option A.
Ma'am,
Sindu.
option A, ma'am.
Jhanavi locked, option A. Jhanavi locked, option B.
Sindhu B, ma'am.
Sindhu locked, option B.
Option A, ma'am.
Option A. May I know your name, please? Kirtana. Okay. Option A. Kirtana locked, option A. So, who all said A? Are you all sure?
Yes, ma'am.
Okay. Who all said B? Are you all confident?
Yes, ma'am.
Okay. Let us see who is confident and who is sure. So the right answer is option A, sorry, option B, euchromatin, which stains very lightly. Option B. So who all answered option B? Congratulations. Who all answered option A? Concentrate on the concept. So give the term for regions in a given structure which are densely packed and stains dark. Option A, heterochromatin. Option B, euchromatin. Option C, pseudochromatin. Option D, none of the above. Who is going to answer first?
Jhanavi A, ma'am.
Jhanavi locked
Kirtana A,
option
ma'am.
A.
Shuchita A, ma'am.
Suchita locked option A.
Tasmia, ma'am.
Tasmia, what is your answer?
B, ma'am.
Sindhu
Tasmia
A, ma'am.
locked option B. Sindhu locked option. Sindhu locked option
A.
A. Okay. Fine. Tasmia, are you sure?
No, ma'am.
Then read the question once again and answer. Kirtana, Pairesh, and Navya.
Option A, ma'am.
Kirtana locked option A. Fine. So the right answer is option A, heterochromatin, which stains darkly. Okay, which stains dark. Then the fourth question goes like this: give the scientific term for bead on string structure. The bead like structures which are there, which we are going to consider as a chromatin. So that you have to say what it is. Option A, chromatin. Option B, nucleosome. Option C, linker DNA. Option D, histone.
Tasmia B, ma'am.
Tasmia locked option B, nucleosome. Yes.
Jhanavi A, ma'am.
Janvi
Sindhu
locked.
A, ma'am.
Okay. Janvi locked option A. Sindhu, what about you?
A, ma'am.
Option A locked by Sindhu. Okay.
Kirtana A, ma'am.
Kirtana A. So the right answer is B, nucleosome. Option B is a right answer. So the fifth question of third main goes like this: in which stage of cell division chromatin is modified into chromosome and seen clearly? Option A, prophase. Option B, metaphase. Option C, anaphase. Option D, telophase. Yes, what is the answer?
Tasmia B, ma'am.
Tasmia locked option B.
Jhanavi B, ma'am.
Jhanavi
Sindhu.
locked
Kirtana.
option B. Kirtana option B. Yes.
Sindhu B.
Sindhu B. All of you locked option B. Are you all confident?
Yes, ma'am.
Okay, then. The right answer is nothing but option B, that is metaphase. In the metaphase, the chromatin will condense and form the chromosome. Okay, thank you for your participation. So you all answered in a mic. Very good. Now, the fourth session, the fourth main, we are going to answer through our messages only like before. So the fourth main question is study the following crime case. So what it is? You have to study the following crime case in which DNA fingerprint of two individuals have been taken. So what is the crime case is? two suspected individual fingerprints are taken, A and B, from their DNA sample, and DNA sample from the crime scene has the fingerprint of C. To answer the following question. Okay, so this is paternal and this is maternal chromosome, and this is a fingerprint they obtained. Okay, now, so suspects A and B obtained from their DNA sample, and DNA sample from the crime scene has the DNA fingerprint C. To answer the following question. Okay, let us see the questions first. The technique to obtain separate bands of DNA fragment is option A, PCR, option D, gel electrophoresis, option C, DNA fingerprint, option D, Sauder blotting. Separate bands of DNA fragment. What is the technique used?
Option B, ma'am. Sindhu.
Answer through the messages, please. Okay, I got the answer. Tasmia said it as option B. Suchita said B. Navya B. Kirtana B. Bhairesh B. Then Janavi B. Then Sindhu also answered B. The right answer is PCR, B, gel electrophoresis, C, DNA fingerprint, and D, Sauder blotting. The right answer is gel electrophoresis to obtain the separate bands. Fine. So here, the next question. The short tandem repeats are present in. The short tandem repeats are present in option A, satellite DNA, option B, any part of DNA, option C, homologous DNA, option D, single strand DNA. Yes. Yes, what is the answer? Janvi said option A, Smita said option A, Bhairesh option A, Tasmia option A, Sindhu option A, Kirtana option A. Fine. So the answer is option A, that is satellite DNA. All of you answered it correctly. Good going. So the next question is, this technique relies on which characteristic of DNA polymorphism? Means on which characteristic of DNA polymorphism this technique is related? Option A, they are variable. Option B, they are inheritable. Option C, they are different from individual to individual. Option D, they are short DNA fragments. So down you will be given the option, that is first option is only A, second option is B, C and D, third option is B and C, fourth option is all of the above. So what is the answer? Bhairesh locked option D, Suchita locked option D, Ruhina option D, then Tasvia option D. Okay. Okay, the right answer is all of the above. Good. Is it right? Compare the VNTR of chromosome number 16 of C with the chromosome number 7 of suspects. C16, chromosome number 16 with chromosome number 7 of suspect. Yes or no? Can we compare chromosome number 16 with chromosome number 7? C is a person who got, means who undergone crime. A and B are the suspect. ಸಿ ಅನ್ನೋ ಪರ್ಸನ್ ಮೇಲೆ ಏನೋ ಕ್ರೈಮ್ ನಡೆದಿದೆ, ಎ ಅಂಡ್ ಬಿ ಅನ್ನೋರ್ ಮೇಲೆ ಡೌಟ್ ಬಂದಿದೆ. ಸೊ ಎ ಅಂಡ್ ಬಿ ಅನ್ನೋರ್ದ DNA ಸ್ಯಾಂಪಲ್ ಕಲೆಕ್ಟ್ ಮಾಡಿದ್ದಾರೆ. ಸೊ ಏನ್ ಹೇಳ್ತಾ ಇದ್ದಾರೆ ಅವರು, ಆ ಕ್ರೈಮ್ ಯಾರ ಮೇಲೆ ನಡೆದಿದೆ ಅಲ್ಲ, ದಟ್ ಈಸ್ ಪರ್ಸನ್ ಸೀಡ್ ಕ್ರೋಮೋಸೋಮ್ ನಂಬರ್ 16 ಮತ್ತು ಸಸ್ಪೆಕ್ಟ್ ಅಂದ್ರೆ ಎ ಅಂಡ್ ಬಿ ಸಸ್ಪೆಕ್ಟ್ ನ ಕ್ರೋಮೋಸೋಮ್ ನಂಬರ್ 7 ನ ಕಂಪೇರ್ ಮಾಡಬಹುದಾ ಅಂತ ಕೇಳ್ತಿದ್ದಾರೆ. ಎಸ್ ಆರ್ ನೋ ಕ್ವಶನ್ ಇಟ್ ಇಸ್. Janvi said yes, Ruhina said yes, Sindhu said yes, Tasmia said yes, Bhairesh said yes. I don't know why all of you are managing your friendship. Once anyone types A, all will type A. Once anyone type yes, all are typing yes. The answer is no, because we have homologous chromosome. Homologous chromosome means what? The chromosomes will be similar in structure and function from person to person. So chromosome number 1 of mine and chromosome number of anyone related to me, they will be similar in shape. Okay, so here we can't compare that. So that is why the answer is no. The red bands of chromosome are obtained at anode pole of
Ma'am,
gel.
but your number are different, no, ma'am.
That is why, is it right to compare VNTR of chromosome number? They ask, is it right? Can you compare chromosome number 16 to chromosome number 7? Will it be similar? If you compare chromosome number 7 to chromosome number 7, we will get to know. If you are comparing number 16 to 7, is it right? They are asking. Because they differ in structure and function, right? Shape also. The question is it right? Did you get my point?
Yes, ma'am.
Yeah. The next question, the red bands of chromosomes are obtained at option A, anode pole of gel electrophoric plate, option B, cathode pole, option C, neutral pole, option D, anywhere of gel electrophoric plate. Yes, what is the answer? Yes, come on. Okay. Tasmia said option D, Kirtana option D, Janavi option D, Sindhu option D. Okay. So the right answer is cathode, that is option B, cathode pole of electrophoric plate. The red bands of chromosome are obtained at cathode pole. We know that the nucleic acids or the nucleotides which forms the genetic material, so those are ionizable. So when they will ionize at positive and negative, the red bands are formed which will go to the cathode pole, which is a positive electrode. These are the negative particles. So the red bands will go at the cathode pole of gel electrophoric plate. Okay, now this is the other kind of question. I want any one of you to read it. Who is going to read? Who is interested? Yes.
Sindhu ma'am.
Okay. Yashaswini, yes, R. Raj. Is it your first and new session? Yashaswini, are you here? Fine. If you're new, I welcome you. Fine. Read following and give answer. Okay, Sindhu, can you read?
Genghis Khan, the Persum Mongolian warrior of the 13th century, may have done more than rule the largest empire in the world. According to a recently published genetic study, he may have helped populate it to an international group of genetics study Y chromosomes genome data number HGP have found that nearly 8% of the men living in the region of the former Mongol Empire carry Y chromosomes that are nearly identical. That translates to 0.5% of the male population in the world, or roughly 16 million
Descendants.
descendants living today.
Okay. Okay, Genghis Khan, the fearsome Mongolian warrior ಅಂದ್ರೆ ಒಬ್ಬ ಹೋರಾಟಗಾರ ಅಂತ ಹೇಳ್ಬೋದು, ಯುದ್ಧ ಮಾಡೋನು. ಆಫ್ ದ 13th century may have done more than rule the largest empire. ತುಂಬಾ ದೊಡ್ಡ ಎಂಪೈರ್ ನ ಅವನು ಹೊಂದಿರ್ತಾನೆ. ಅಕಾರ್ಡಿಂಗ್ ಟು ಅ ರೀಸೆಂಟ್ ಪಬ್ಲಿಷ್ ಜೆನೆಟಿಕ್ ಸ್ಟಡಿ ಮಾಡಿದಾಗ ಆ ಎಂಪೈರ್ದು ಹಿ ಮೇ ಹ್ಯಾವ್ ಹೆಲ್ಪ್ ಪಾಪ್ಯುಲೇಟ್ ಇಟ್ ಟು. ಸೊ ಆ ಎಂಪೈರ್ ನ ಅವನ್ ಅಂಡರ್ ಏನ್ ಎಂಪೈರ್ ಬರುತ್ತೋ, ಏನ್ ಸಂಸ್ಥಾನ ಬರುತ್ತೋ, ಸೊ ಆ ಸಂಸ್ಥಾನನ ಅದರಲ್ಲಿ ಪಾಪ್ಯುಲೇಷನ್ ಜಾಸ್ತಿ ಮಾಡೋ ಹಾಗೆ ಅವನು ಹೆಲ್ಪ್ ಮಾಡಿರ್ತಾನೆ ಅಂತ. ಹೇಗೆ? an international group of genetic scientists studying Y chromosome genome data related to that empire under HGP, okay, have found ಏನ್ ಫೈಂಡ್ ಮಾಡಿದ್ದಾರೆ ಅಂದ್ರೆ 8% of the men living in the region. So 8% of the men means male person, okay, who is living in the region that is in Mongol Empire carry Y chromosome that are nearly identical. ಅಂದ್ರೆ ಒಂದು ಸ್ವಲ್ಪ ಜನನ ಮೇಲ್ ಜೆನೆಟಿಕ್ ಜೀನೋಮ್ ನ ಸ್ಟಡಿ ಮಾಡಿದಾಗ ಅವ್ರಿಗೆ ಏನ್ ಕಾಣಿಸಿದೆ, ವೈ ಕ್ರೋಮೋಸೋಮ್ ಏನ್ ಸ್ಟಡಿ ಮಾಡಿದಾರಲ್ಲ, ಸೊ ಆ ವೈ ಕ್ರೋಮೋಸೋಮ್ ರಿಲೇಟೆಡ್ ಆಗಿದೆ ಮತ್ತು ಐಡೆಂಟಿಕಲ್ ಸೇಮ್ ಶೇಪ್ ಮೀನ್ಸ್ ಸಮ್ವಾಟ್ ಸಿಮಿಲರ್ ಇದೆ ಅಂತ. we know that the blood relations, for example my mother, my father, my daughter, so here we can compare our chromosome, the structure or shape will be quite identical, right? So that translates to 0.5% of the male population in the world, or roughly 16 million descendants living today. So ಏನಾಗಿದೆ, ಅದು ಒಂದೇ ಐಡೆಂಟಿಕಲ್ ಆಗಿರೋದ್ರಿಂದ, ಅಷ್ಟು ಐಡೆಂಟಿಕಲ್ ವೈ ಕ್ರೋಮೋಸೋಮ್ ಬಂದಿರೋದ್ರಿಂದ ನಮಗೇನಾಗತ್ತೆ, ಆಲ್ಮೋಸ್ಟ್ ಆಲ್ ಅಲ್ಲಿ ಏನ್ ಪಾಪ್ಯುಲೇಷನ್ ಇದೆ ಅದು ಒಂದೇ ಮನೆತನದ್ದು ಅಥವಾ ಒಂದೇ ರಿಲೇಷನ್ಸ್ ತುಂಬಾ ಜನ ಆಗಿ ಹಾಗಾಗಿರ್ಬಹುದು ಅಂತ ಅನ್ಸುತ್ತೆ. ಸೊ ಅದಕ್ಕೆ ಏನ್ ಕೇಳ್ತಿದ್ದಾರೆ, ಸರ್ಟನ್ ದೇ ಹ್ಯಾವ್ ಆಸ್ಕ್ಡ್ ಸರ್ಟನ್ ಕ್ವಶನ್ based on this study which we have to read. Okay, question ಅರ್ಥ ಆಯ್ತಾ ಎಲ್ಲರಿಗೂ?
Yes, ma'am.
Okay, ಎಲ್ಲರಿಗೂ ಅರ್ಥ ಆಯ್ತಾ?
Yes, ma'am.
Yes, ma'am.
Let us see the question. The first question goes like this: identify the phenomenon mentioned above. ಮೇಲ್ ಕೇಳಿರೋ ಫಿನೋಮಿನನ್ ಯಾವುದು ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಆಪ್ಷನ್ ಎ human genome project, option B density gradient separation, option C radioactive marking, option D heavy isotope marking. So yaudadu? You have to message me your answer, please. Tasmia said option A. Janvi marked option B. Kirtana option B. What about others? Suchita option B. Sindhu option B. Ruhina option C. The right answer is option A, as it is related to the population and the genome of a specific community or specific region, people related to the region. The right answer is option A, that is human genome project. The first correct answer is given by Tasmia, option A. Good. The next question is, which chromosome has fewest gene? First chromosome, B X chromosome, C Y chromosome, D third chromosome. ತುಂಬಾ ಕಡಿಮೆ ಜೀನ್ ಇರೋ ಕ್ರೋಮೋಸೋಮ್ ಯಾವುದು ಅಂತ ಕೇಳಿದ್ದಾರೆ. So what is the answer? Yes, Bhairesh said option A. Janvi said option C. Sindhu option C. Then Tasmia option C. Navya option C. Ruhina option A. Shuchita option C. Okay. Kirtana option C. Fine. Okay. So the correct answer here is given by most of you, that is option C. So when we study, okay, the chromosome, so we know that the X chromosome, when we study, it will be like this, and Y chromosome, when we study the XY, the chromosomal complement of a male. So when you see the diagrammatic representation of a Y chromosome, it is smaller in size. So smaller in size means what? It will be having fewer genes. So option C is the right answer. Good. The third question is, which chromosome has most genes? Which chromosome has most genes? Option A, X, option B, Y, option C, first option D. 1 minute. Second. Yes. What is the answer? Fine. Tasmia said option C, Kirtana option C, Janavi option C, Sindhu option C, Ruhina option C, Janavi option C. Okay. Then, yeah. So the right answer here is fifth one, fifth main third one. So option C, that is a first chromosome. Option C is a right answer. First chromosome will be having many genes. Major approaches involved in methodologies of HGP, Human Genome Project. Option A, expressed sequence tags, sequence annotation. Option B, sequence annotation. Option C, both A and B. Option B, none of the above. So what is the answer? Yes, may I know the answer? Tasmia log option B, Janvi option B, Kirtana option B, Smita option B, Ruhina option C, Shuchita option B. Yes. Anyone? Sindhu option C. So major approaches involved in methodologies of HGP. So here fourth one, option B, that is sequence annotation. Option A, expressed sequence tags. So both are interrelated. That is why both are involved in methodologies of Human Genome Project, right? So option C is the right answer. That is both A and B. Both A and B is a answer. So almost all of you answered C. Sindhu answered, Ruhina, then yeah, okay, fine. So the next question goes like this, just a minute. Which is not a goal of HGP, that is Human Genome Project? Option A, identify all the approximately 20,000 to 25,000 genes in human DNA. Option B, determine the sequence of the 3 billion chemical base pairs that make up human DNA. Option C, store this information in databases and improve tools for data analysis. Option D, inhibition of transfer related technologies to other sectors such as industries. What is your answer? Fast. Bhairesh option D. Janavi option B. Yes. Tasmia option B. Suchita option D. Sindhu option B. Kirtana option B. Fine. So the right answer is inhibition of transfer related technologies to other sectors such as industry. So whatever they find in human genome project, okay, so whatever they find in human genome project, so it is not shared to other industries for their benefits, okay, it is maintained secretly. So inhibition, what is the main rule? Inhibition of transfer related technologies to other sectors such as industry. So option D is the right answer. It is given by Shuchita, then Bhairesh. Okay, both of them given option D, that is correct answer. Good going. So this is regarding certain kind of questions which might be asked in the upcoming exam. Okay, now fine. Just a minute. So yesterday we were discussing molecular bases. So we discussed up to I guess 22nd, then 23rd question. Okay, fine. So we discussed until 20, I guess, in two conservative same chains. Okay. The term used by Mendel, this we discussed. Identify the incorrect statements, this we discussed. RNA is liable, this we discussed. Which of the genetic material, this we discussed. DNA is structured due to presence of uracil, this we discussed. The first genetic material. Okay, from 26th we have to discuss. Okay, so let us start from 26th question. Okay, now the question is, the first genetic material is option A, protein, option B, for carbohydrate, option C, DNA, option D, RNA. Okay, the first genetic material which is discovered, what is the answer? Fine. Smita, yes, option C. Kirtana, option C. Janvi, option C. Then, okay, Tasmia, option D. Yes, Sindhu, option C. Navya, option C. Fine. Good. So the right answer is, the first discovered genetic material is actually RNA. So the right answer is option D. Okay. Now the next question. What is hnRNA? A, human RNA, B, hypernuclear RNA, C, heteronuclear RNA, D, heterogeneous nuclear RNA. 27th question. Yes, come on. What is the answer? Tasmia said option D. Janvi said option D. Smita, option D. Suchita, option D. Yes. Kirtana, option D. Sindhu, option D. Okay. So the right answer for this question, that is 27, is heterogeneous nuclear DNA, that is hnRNA. So all of you, hnDNA, all of you answered it correctly. That is option D. Good. Then the next question is, what will be the effect of this experiment on rat? A, rat coat becomes smooth. B, rat dies. C, rat is unaffected. D, rat body show phagocytosis and no pneumonia. So what happens here? Living S cells, smooth strained cells is injected into the rat. So when smooth strain cell is injected, what will happen to the rat, they asked. So what is the answer? Be fast. Janvi, option B. Okay. Sindhu, option B. Okay. Sindhu, option B. What about others? Smita S, option B. Tasmiya, option B. Kirtana, option B. Okay. Okay. So the right answer is rat dies, that is option B. When smooth strained cells, so they will cause pneumonia and the rat dies. So that is the answer. The right answer is option B. Most of you answered it correctly. The promoter site and terminator site for transcription are located at 3' downstream, 5' upstream end respectively of transcription. That is 3' to 5' end anta. 5' upstream, 3' downstream, that is 5' to 3' end. 5 upstream and 3' downstream end. So what is the right option? Promoter ಮತ್ತೆ terminator site ಎಲ್ಲಿ ಲೊಕೇಟ್ ಆಗಿರ್ತವೆ? Tasmiya said option B, Kirtana option B. Then yes, sorry, what about others? Janvi said option A, Shuchita said option B. Sindhu said option A. Okay. Okay. So the right answer is given by Shuchita, then Kirtana, Tasmiya. Fine. So the right option is option B, that is 5' end to 3' end. With regard to mature mRNA in eukaryotes, which of the following is true? Option A, exons and introns do not appear in mature RNA. Option B, exons appear but introns do not appear in mature RNA. Option C, introns appear but exons do not appear in mature RNA, mature mRNA. Option D, both exons and introns appear in the mature RNA, mature mRNA. So what is the answer? Yes. What is the answer? Tasmiya said option B. Janvi said option D. Sindhu said option D. Navya said option B. Kirtana said option B. The right answer is exons appear but introns do not appear. So in the mature mRNA. So the right answer is option B. Option B. 31st question. If the number of base pairs in double-stranded DNA is 200, the number of adenine is 60, then what will be the number of guanine? Option A 60, option B 40, option C 80, option D 100. Yes, adenine 6, total DNA 200. ಅದ್ರಲ್ಲಿ ಅಡಿನೈನ್ 60 ಇದ್ರೆ ಗ್ವಾನೈನ್ ಎಷ್ಟಿರುತ್ತೆ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಸೊ ವಾಟ್ ಈಸ್ ದ ಆನ್ಸರ್? Ruhina said option B, Janvi said option A, Sindhu said option A, Tasmia said option B, Smita said option A, Kirtana said option B, the right answer is option B, answered by Kirtana, Tasmia. Okay, answered by Kirtana and Tasmia and Ruhina. So here what happens? Adenine is 60 means guanine will be 40. Adenine is 60 means thymine will be 60. 60 plus 60, 120. 40 plus 40, 80. 120 plus 80, 200. So like that. Then discontinuous synthesis of DNA occurs on one strand because option A, DNA molecule being synthesized is very long. Option B, DNA dependent, that is DNA polymerase catalyzes polymerization in one direction. Option C, it is a more efficient process. Option D, DNA ligase has to have some role. So what is the answer? DNA discontinuous means discontinuous strand occurs on one side. Why? Why it occurs on one side? Option B. Tasmia said option B. Sindhu option B. Then yes. Kirtana said option B. Janvi said option B. Okay. So the right answer is option B, that is DNA polymerase only acts on 5' to 3' direction. That is why the discontinuous synthesis will be formed in the other strand. Okay. Now, so the following are the functions of RNA. So fine. I will take just 2 or 3 minutes break. I'm going to drink water. And you also take a break if you want. Otherwise, study the following questions and be ready to answer. Okay. So let us meet 2 to 4 after 2 minutes. So I have noted down the participant's name. Please make sure to continue in a session if you want the MCQs based on this, what we discussed today. Okay. Right. Okay. Okay, I think you have read the question. That is 33rd question. Which of the following are the functions of RNA? So that is option A, it is carrier genetic information from DNA to ribosome synthesizing polypeptides. Option B, it carries amino acids to ribosome. Option C, it is constituent components of ribosome. Option D, all of the above. So what is the answer? Tasmia said option A. Janvi said option D. Then what about others? Suchita said option D. Sindhu said option D. Kirtana also voted for D. Okay, the right answer here is, all of the above, because of these RNA function. Okay, these all are the functions of RNA, right? Okay. 34th question. RNA polymerase 2 is responsible for the transcription of option A, tRNA, option B, rRNA, option C, hnRNA, option D, snRNA. Yes. What is the answer? Janvi said option C. Tasmia said option D. Yes. Tasmia said option D. Navya said option C. Kirtana said option C. Yes. Two minutes, I will be back. Students, I apologize. I have got some emergency. I will be continuing the session tomorrow. Sorry for that.
Thank you, ma'am. Thank you, ma'am.
So